Neurons and synaptic activity. Just a reminder that this is what a neuron looks like. Up here we have the dendrites, and you can see connections. The dendrites, their role really is to collect information from other cells. So these would be synapses from other cells that you see coming in. And we have that information is integrated in the cell body, also known as soma, and then transmitted down the axon to the terminal, different terminal types, where it's going to synapse. These terminals will synapse with perhaps dendrites of additional neurons. So the axons collect the information, the dendrites collect the information, cell body integrates the information, and the axon sends that information. Flow is from this in that direction. A reminder too that the membrane of a neuron is a phospholipid bilayer. So it keeps certain substances outside and other substances inside the neuron. Although there are proteins that traverse this membrane, and we'll talk about some of those. The anatomy of the neuron we discussed, and it's been covered in the book. So we have dendrites, cell body. The axon hillock is the beginning point where the cell body becomes the axon. These are myelin sheaths that speed up neural transmission so that the action potential can jump from node to node. And then we have our terminal. And a reminder of the action potential. At rest, the inside of the neuron is very negative, minus 70 millivolts, and that's because negatively charged anions are stuck inside the neuron, inside the membrane. And what letter C is here is the threshold, about minus 50 millivolts. And when threshold is reached, the sodium channels will open up, and sodium, what's happening in A is sodium is rushing into the neuron, causing this peak of about plus 30 or plus 50 millivolts. And at the same time, there's potassium that's inside the axon that's leaving. So the effect that's being felt in part B is the potassium ions leaving. Potassium and sodium are both positively charged, so because similar charges repel each other, potassium is repelled out of the axon and is flowing outside into the extracellular fluid. So much leaves that at point C here we have what's called a refractory period. You see how it's more negative now inside the neuron than it is at start, at rest. It's called the refractory period. And at this point there's going to be a pump that will pump the sodium ions out of the neuron and pump the potassium ions back into the neuron. So that's what's happening down here, this upswing, and then it returns the axon to resting potential, which is letter D. And all this takes only 2 to 3 milliseconds each action potential. But that's why having the nodes of Ranvier is helpful, because you don't need to have an action potential here, 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 here. It's able to jump from the beginning to this one, to this one, to this one. That makes it go a lot faster. So in this class we're talking quite a bit about really the neurotransmitters. So neurotransmitters are often packaged in the cell body and then transferred via the microtubules in the axon down to the terminal, where then ultimately they'll be released. So more on that. There are different ways to interact with the lifetime of a neurotransmitter. So we're going to first go over the lifetime of the neurotransmitter. First of all, we have synthesis, where neurotransmitters are created from their component substances. They're going to be stored in vesicles where they're safe. They're not safe outside of a vesicle because there are enzymes that degrade them. And number 3, there's a release caused from an action potential. Remember, the action potential comes down the axon here. That opens up calcium channels, calcium, which causes the vesicles to move and fuse with the membrane. And through a process called exocytosis, the neurotransmitter then is spilled into the synapse. So that's number 3, release. There will be a receptor interaction. Could be with ionotropic or metabotropic receptors. There's an inactivation, maybe through enzymes that deactivate the neurotransmitter. And then there's a reuptake mechanism. Possible for degradation, again, if the neurotransmitters aren't stored in synaptic vesicles. So we're going to go through each of those in a little bit more detail. So first of all, we have synthesis. That's the formation of the neurotransmitters. And there are certain types of precursors that create neurotransmitters. These precursors are brought into the neuron via the bloodstream. They're taken up either by the cell body or the terminal. And these precursors are often substances that come from our diet. So foods that we eat help us make neurotransmitters. And there are enzymes that help put the ingredients together. If you look on this diagram here, right now we're talking about synthesis, kind of putting these neurotransmitters together via the help of synthesizing enzymes. And they're different for each neurotransmitter, for a lot of the neurotransmitters. Next, the neurotransmitters are stored in vesicles. Each vesicle contains approximately the same amount of neurotransmitter, and they're in these vesicles for protection, because if they're not in a vesicle, they may be attacked by degrading enzymes. Then there's the third phase is the release. Action potential causes the vesicles to fuse and synthesize, and through a process of exocytosis, release the neurotransmitter into the synapse. Now they're going to have a binding to the receptor, and that binding can be on an ionotropic receptor. You can see this neurotransmitter fits perfectly in this receptor, and that causes the ion channel to open up and to let ions into this postsynaptic cell. So up here is the synapse, down here is the postsynaptic cell. And if these ions are sodium, which has a positive charge, that would cause an excitatory postsynaptic potential. If the ion is chloride ions, that's negatively charged, that would cause inhibitory postsynaptic potentials. So these fluctuations bring this next cell closer to, like excitatory, or farther away, like an inhibitory, firing its own action potential. The other type of receptor are metabotropic receptors, and here a neurotransmitter will bind to a metabotropic receptor, which causes secondary effects, a second messenger system, to be activated in the postsynaptic cell, which ultimately may open up an ion channel or cause other changes in this postsynaptic cell that it will alter its firing rate, to either increasing or decreasing the number of action potentials it's sending. All right, so once you've made the neurotransmitter, it's been stored in the vesicles, it's been released via an action potential, it's had its effect on the postsynaptic cell, now it's time to get rid of, you've got to stop that message. This message is very brief. So you're either going to metabolize it with an enzyme that's found in the synapse or on the postsynaptic cell, or you might reuptake, use a carrier protein that will bring the neurotransmitter back into the presynaptic cell. Or in one case, glutamate, one neurotransmitter we'll talk about, there's a reuptake by a nearby glial cell that we'll discuss a little bit later. This diagram also shows something called autoreceptors. This is another type of membrane protein that helps regulate how much neurotransmitter is created and released. If the autoreceptors detect that too much neurotransmitter is in the synapse, the autoreceptor will cause changes in this presynaptic cell so that to decrease the production and synthesis of the neurotransmitter. So it's a way to regulate neurotransmitter release.